In a quiet laboratory, scientists built something that blurs the line between chemistry and life. It wasn't born, it wasn't evolved, and it wasn't designed to live. Yet somehow, it behaved like something alive. Here's where it gets unsettling. Because no DNA told it what to do. No genes, no instructions, no evolutionary history. And still, it organized itself. That shouldn't happen according to classical biology. Life, we were told, needs instructions. But this system didn't follow a script. It forced scientists to ask a dangerous question. What actually separates life from non-life? Because if the boundary is thinner than we thought, then creating life may be easier than destroying it. This is the experiment forcing science to redefine what alive really means. To understand why this shook scientists, we first need to answer a basic question. What do we actually mean when we say something is alive? Most biology textbooks give a checklist: metabolism, growth, response to the environment, and the ability to maintain internal order. Living cells constantly fight against disorder, like a spinning plate that has to keep moving to stay upright. Stop the motion, and the system collapses. For decades, scientists believed DNA was the instruction manual behind all of this. DNA tells cells what proteins to make and when. Those proteins build structures, process energy, and keep cells alive. So life was defined as chemistry plus instructions. Remove the instructions, and biology should stop. That assumption shaped everything from medicine to evolution. But it also hid an uncomfortable possibility. What if order can emerge without instructions? In physics, this idea isn't new. Snowflakes, hurricanes, and galaxies all self-organize. No blueprint tells them what to become. They follow simple rules, and complexity emerges. Biology, however, was thought to be different. Life was supposed to need genes, until researchers tried removing as much as possible. Not to create life, but to study its limits. That decision led directly to the experiment that shocked biology. This is the experiment forcing science to redefine what alive really means. This story didn't begin with a plan to create life. It began with scientists trying to understand how life might have first emerged. Specifically, how simple chemistry could turn into organized biology. The work happened at the edges of synthetic biology, where researchers build stripped-down versions of cells, not to replace life, but to understand it. They asked a simple but risky question. What is the minimum needed for a system to behave like life? To answer it, they began removing almost everything. No nucleus, no DNA, no genes telling the system what to become. What remained was chemistry, membranes, and energy. The expectation was clear. The system should do nothing. At best, it might briefly assemble, then fall apart. That's what decades of biology predicted. But when energy was introduced, something unexpected happened. Instead of collapsing, the structures stabilized. And that was the first moment scientists realized they might be watching something new. At first, the researchers assumed the stability was temporary. Chemical systems can briefly look organized before falling apart. So they waited. Minutes passed, then hours. The structures didn't collapse. They maintained boundaries and regulated what crossed in and out. That alone was unexpected. But then the system began responding to changes. Increase energy and activity increased. Reduce it and the system slowed. Not randomly, but predictably. That predictability changed the interpretation completely. Because predictability suggests regulation. And regulation is a hallmark of living systems. The system wasn't alive, but it wasn't non-living either. It occupied a space biology had never clearly defined. Some researchers called it lifelike. Others preferred something more cautious. But everyone agreed on one thing. This behavior wasn't programmed. It emerged on its own. When the results were shared, the reaction wasn't excitement, it was caution. Because biology has learned to be careful with surprises. Independent teams began testing similar systems, not to prove life was created, but to test the behavior. And the same patterns kept appearing: self-organization, regulation, response to energy, all without genes telling the system what to do. That forced biologists to reconsider a basic assumption: that instructions must always come first. Instead, behavior might emerge first and instructions later. This has implications far beyond origin of life research. In medicine, it hints at new ways to stabilize failing cells. In synthetic biology, it changes how systems are designed. Instead of controlling every step, scientists may guide conditions, like tending soil instead of programming every leaf. Artificial life research also took notice, because learning and organization may not require intelligence, just the right rules, energy, and feedback. That realization unsettled some researchers, because it blurs the line between building and creating, and raises questions science doesn't usually ask. If lifelike behavior emerges easily, how careful do we need to be? And who decides when something crosses the line? Those questions push this science beyond labs and into responsibility. Before anyone talks about applications, scientists pause to ask what could go wrong. Because creating systems that organize themselves isn't inherently safe. The same rules that create order can also amplify instability. Without limits, feedback loops can spiral out of control. That's why these systems are studied under strict containment. No release, no replication outside controlled environments. The goal isn't to create new life forms. It's to understand how complexity emerges from simplicity. That knowledge could help prevent diseases linked to cellular breakdown, or design safer synthetic systems that fail gracefully. But only if society sets boundaries before capability races ahead. Because once something is easy to build, it's hard to unbuild. That's why transparency matters as much as innovation. And why ethics isn't a brake, it's a steering wheel. This discovery doesn't redefine life. It reveals how close matter already is to becoming organized. If you want to understand the discoveries reshaping our future, subscribe to Behind the Breakthroughs.